Okay, thanks. And thanks for the invitation. I do have 30 minutes. We're starting 15 minutes late. So it's going to go that long. So thanks for the opportunity to talk here. I'm going to kind of continue on the theme of induced pluripotent stem cells, but more specifically how we've used these cells to derive lymphocytes, in particular natural killer cells, which can be used for cell therapies. And so just my disclosures, I collaborate with Fate Therapeutics, who I think's already talked about some of this work at this meeting. So this is a figure from a nice review from Crystal Mackel talking about CAR T-cell therapies, which again I think folks at this meeting should be very familiar with. This has been really a great advance. It's really gotten a lot of increased interest in use of cell-based therapies, though as somebody who does clinical hematology and bone marrow transplant, I'll remind you we've actually been doing cell therapy successfully for 50 years, since the late 60s, so this did not start with CAR T cells, but bone marrow transplant really is cell-based therapies, and we know a lot of the effectiveness, especially for hematologic malignancies, is through graft versus leukemia activity from the donor lymphocytes. But now being able to do this on a sort of more precise fashion, and this is really extended, and this is really nice showing all the trials that are going on, but the continued issues with the current CAR T-cell therapy. There's antigen escape, there's toxicities from the therapy, high cost, so-called financial toxicity, and the time that it takes to prepare these cells. So this is done on a patient-specific basis. The cells have to be collected. It takes about 21 days to make the cells, given back to the patient. Not every patient has that much time, plus the time to get screened. And I know from personal experience that we've had patients who don't have 21 days or more for this process. So while it's been a great success, there's certainly rooms to improve. And so really, I like this summary of my idea of really where we want to go for cell-based therapy for cancer. That is, we want to make cells as easy to use as drugs. That is, they can be targeted like CAR T cells are, but to even further improve the on-target effects and reduce off-target toxicities. But I think we really want this to be standardized and so-called quote-unquote off-the-shelf. That is, we want every patient to get the same thing. Again, you hear so much about personalized medicine, which is great, but I don't want to treat one patient at a time. I want to develop therapies that we can treat hundreds of patients, just like drugs do now. And that's more economical. They're ready to go, and when the patient needs them, they can get these cells. So the way that we've gone about doing this is by use of human pluripotent stem cells. This is from an early review a decade ago now, where we've used both, starting with human embryonic stem cells and now with induced pluripotent stem cells to study blood cell development. And there's a variety of methods that can be used to differentiate these cells. My original interest was to use these to derive hematopoietic stem cells that could be used for transplantation. If anybody's interested in doing that, really think hard about doing that, because it's really hard. And despite 20 years of trying to do that with human cells and 40 years of people trying to do that with mouse cells, nobody's actually really been successful unless you genetically modify those cells in some fashion. But what we have really gotten good at is we can make more differentiated cells. If you think of these as sort of a developmental slope or pathway, the cells want to go downhill. So we've really focused now on production of lymphocytes, particularly natural killer cells. Other groups have done red cells, platelets, monocytes, and so on. So this is just your one slide reminder of what natural killer cells are and why they're important. These are part of your normal immune system. They're known to kill virally infected cells and tumor cells. They do this as so-called innate immune cells. That is, they can do this without prior sensitization and without HLA restriction. So I'm not going to go through the details of how NK cells are regulated. They have repertoires of activating and inhibitory receptors, and these are different than the T cell receptors that require self-HLA. So what this means is that NK cells work normally as allogeneic cells. My NK cells can kill your tumor cells, and vice versa. They don't have to be matched to a specific patient. And we've been doing clinical trials of NK cells for 10 to 15 years, actually largely led by my former colleague when I was at the University of Minnesota, Jeff Miller, who's been doing lead trials using NK cells from haploidentical donors, largely to treat AML. These have been very effective. About 30 to 50% of patients can get into remission. And other groups, WashU, Boston, and so on, have continued these type of trials. However, again, those are a donor-specific product, which, while effective and a few hundred patients have been treated, not as many patients probably as could benefit. What we do know from those trials using allogeneic NK cells, even though these are allogeneic cells, they don't cause graft versus host disease, they don't cause cytokine release syndrome, and they don't cause the neurotoxicity that's been seen in the CAR T cell therapy. So this is how we make the NK cells from the human pluripotent stem cells. Other groups do this different ways. We, as Irene mentioned, also use this so-called SPIN-EB method. These are all serum-free, stroma-free, defined conditions. You really just spin the cells, 3,000, 5,000 cells per well in a 96-well plate with a cocktail of hematopoietic cytokines, and what you get is actually hematopoietic organoids. Which is probably a better term than these EBs. They contain hematopoietic progenitors as seen by these CD34, CD43, and 45 positive cells, but they also contain endothelial cells and stromal cells. So we actually found we could just take this whole organoid, put it down on plastic, again with the right cytokines. The NK cells will actually develop and expand on top of those stromal cells. So we get this nice population of CD56 positive cells, which is the typical marker for NK cells. We don't get myeloid cells. And then if we want to scale these up further, we can use what many groups have done is an irradiated so-called artificial antigen presenting cell. This is an engineered K562 cell that in the version we use expresses IL-21, 41BB, and some other stimuli, and you can really maintain these cells for months at a time and expand these into clinically suitable numbers. If we look at the phenotype and the genotype of these ES and iPS-derived NK cells shown here compared to NK cells from peripheral blood or from cord blood, they're all very similar. So you get nice CD56 positive populations. They, in varying degrees, express the so-called KIRs, killer immunoglobulin receptors. They express CD16, which we'll talk about more, and they express the activating receptors like NKG2D, P44, P46, and so on. We can look at the gene expression. Again, the ES and iPS-derived NK cells have gene profiles very much like peripheral blood and cord blood-derived NK cells, even more closely matched if you specifically hone in on the gene expression for NK-mediated cytotoxicity pathways. So again, these look like typical NK cell populations. We then tested these first in vitro. We saw that they could kill a variety of tumor cells in vitro. This is the first test we did in mice. We injected immunodeficient mice with luciferase expressing leukemia cells. Sub-Q, we gave them just one IV injection of, in this case, ES-derived NK cells, or at the time we were using cord blood-derived NK cells. The cord blood-derived NK cells, you got some anti-tumor activity, but with the ES-derived cells, just one injection, and then the mice get cytokines for 3 weeks afterwards to keep the human NK cells happy and healthy. That was enough to completely eliminate these leukemia cells in vivo. Every mouse that we treated, 13 out of 13 mice, were successfully treated. So this got us interested is, could we start to move this towards clinical therapy, and what else could we do with these cells? We subsequently tested these in a bit more challenging model. As I mentioned, most of the activity clinically has been seen for NK cells against AML. There have been trials against solid tumors, which have shown some activities, but less so than the AML cells. So this is looking at an ovarian cancer model where the ovarian cells are given IP and the NK cells are given IP. Again, if we compare to peripheral blood NK cells, you get some anti-tumor activity. The iPS-derived NK cells gave us better anti-tumor activity, but unlike the leukemia cells that could be cleared with just one dose of NK cells, this shows sort of the tougher barrier for the solid tumors, is that, well, we could get remissions and we could potentially give multiple treatments with just one injection kind of in parallel to the leukemia cells. We could get remission, but the tumors came back. So we wanted to use this and see how could we now not only make NK cells with anti-tumor activity against AML, how could we use these as a platform to improve their activity against solid tumors? And I'm going to talk about some of these strategies. The first one was published last year on engineering iPS-derived NK cells with novel CARs. I'll talk a bit then about CD16 expression, which mediates improved ADCC, as well as some genetic modifications we can use to improve the activity of these cells. So the first strategy is to produce CAR expressing iPS-derived NK cells. And when we do these genetic modifications, you need to remember that we're doing all these modifications on the stem cell level. We don't wait for the cells to differentiate and gene modify those. If we can do all the engineering in the ES or iPS cells, what we've shown is that we can then, make the CAR expression in the iPS cell, differentiate them into NK cells, and we get CAR expressing NK cells. And again, the advantages is we get a much more homogeneous starting cell population. You can use any of a variety of methods to gene modify ES or iPS cells. We actually do most of the work in our lab using transposons, either Sleeping Beauty or piggyback, depending on the size of the vectors. Others have used retrovirus, lentivirus, and so on. They all work pretty efficiently. And then if you want, you can target the insertion to a conserved site. And really, as I'll show you, you can then differentiate these cells and get a very uniform population, essentially 100% CAR expressing cells. So we initially did this with a T-cell CAR construct. We had against both anti-CD19 for B-cell malignancies as well as anti-mesothelin, which is expressed in a variety of solid tumors. We used a so-called third-generation CAR T-cell construct. We put this into the ES cells. This shows you that we can get CD56 positive CAR expressing cells, 93% of our cells. They had a typical phenotype. They worked great in vitro, and then when we tested them in vivo, as I'll show you in a few slides, they didn't work at all. They were actually no better than the unmodified iPS-derived NK cells if we use this construct in vivo. And so we kind of went back to the drawing board, and we actually started to design CARs specifically for NK cells, because even though there's a lot of conserved signaling pathways between T cells and NK cells, we thought, well, maybe we can improve upon this. And so we designed and tested about a dozen different CAR-specific NK cells with different transmembrane domains, intracellular domains, all with the same CD3 zeta signaling domain, which is also expressed in NK cells as part of the CD16 complex. And the short answer that we get to is the so-called CAR4 construct, which has an NKG2D transmembrane domain, 2B4 intracellular domain, and CD3 zeta. We use this against anti-mesothelin. And the way we identified this construct is we actually screen these in NK92 cells. This is an NK cell line. It grows very easily. It's easy to genetically modify. And we tested these against 2 mesothelin negative and 2 mesothelin positive tumor cell populations. And what we found is, if you see sort of the pointers don't work great, but in C and E, that 3 of the CAR constructs gave us the highest activity. In E, there were 2 kind of intermediary. And what these had in common, these are actually so-called CARs 4, 7, and 9. They actually all had the same 2B4, NKG2D, 2B4 zeta domains. 7 and 9 just added 1 additional domain, either DAP10 or 41BB. We figured those weren't really adding anything, so we just stuck with the simpler construct. We then put this into our iPSCs, made iPS-derived NK cells that express this CAR4. You can see we get a nice CD56 positive population. We're now using GFP to track our CAR expression. They're all GFP positive. They express, again, the typical NK cell activation receptors. We tested these in vitro. They look good, and then we did these in vivo. And again, this is now using the same ovarian cancer model. They get IP ovarian cells. 4 days later, they get 1 dose of NK cells plus cytokines for 3 weeks. No treatment, the tumors grow. The next 3 lines, either using peripheral blood NK cells, unmodified iPS NK cells, or this actually shows if you use iPSCs expressing that T cell CAR construct is exactly the same as the unmodified iPS NK cells. But if we put in our CAR4, we actually got much deeper remissions and better survival. And this is what's shown here is, again, in green is the CAR4, gave us better remissions and significantly improved survival. And we were pretty happy with this, and then the reviewers kept asking us this question, is how do the iPS and CAR NK cells compare to so-called conventional CAR T cells? And despite us saying that's not really the point, even if these were the same, we think there's enough advantages of being able to do this from iPSCs as a standardized cell product that, you know, we weren't thrilled, but we bit the bullet, made CAR T cells, and we were actually surprised, and this worked out in somewhat of our favor. So we did this study. We actually made, again, the conventional CAR T cells, which are in yellow, compared to our CAR iPS NK cells. We actually did the opposite things. We put the T cell CAR in NK cells and the NK CAR in T cells, which are sort of the intermediary lines. These are in vitro cytotoxicity assays. What we found is the CAR NK cells killed faster and more completely than the CAR T cells, which were in yellow. And then we tested these in vivo. And again, so if we have tumor alone, tumors plus unmodified T cells, the CAR T cell and the T cell, if you go out to day 21, is giving some nice anti-tumor activity. The unmodified iPS NK cells, not much activity, and then in the bottom is the CAR NK cells. If you look out to day 21 on sort of panel D there, the anti-tumor activity between the CAR NK cells and the CAR T cells is really very similar. But when the kinetics is different. The CAR NK cells actually killed faster, and then you kind of get a plateau. The CAR T cells take a little time to warm up, but then once they get going, they start killing the tumor quite well. But what we found is that the mice that were getting the CAR T cells were actually looking sick and starting to lose weight. And that's shown on the bottom right panel. And what happened is that we found that the mice that got the CAR T cells actually started to have increased proliferation of their CARs up to, we don't show here, 20-90% of the cells in the peripheral blood became CAR T cells, and they express high levels of these inflammatory cytokines, interferon, TNF alpha, and IL-6, which are in yellow. The CAR NK cells had some of this, but at much more modest levels. And so what we're actually seeing in the mice treated with the CAR T cells is some combination of cytokine release syndrome and graft versus host disease that we don't see with the CAR NK cells. So again, even though the CAR NK cells are allogeneic, or in this case Zealand genetic cells, you don't get graft versus host disease. And so the mice from the CAR T cells actually started to die not from tumors, but from the toxicity. And that's shown here in the yellow, whereas the mice that got the CAR NK cells actually survived longer. So those reviewer concerns actually worked in our favor. We could show not only are the CAR NK cells just as effective, they're potentially safer than the CAR T cells. So now that's all been published last year. A couple stories that have just been submitted. One is on CD16 and one is on some internal gene edits. So this story on CD16, so CD16 is the FC receptor. It binds antibody, it mediates so-called antibody-dependent cell-mediated cytotoxicity. So when you give patients monoclonal antibodies like rituximab and cetuximab, a lot of their activity is through NK cell-mediated ADCC. However, what I learned relatively recently is that when NK cells get activated, CD16 is actually cleaved off the surface of the NK cell by a metalloprotease called ADAM17. With our collaborator Bruce Walczek at University of Minnesota, he identified the cleavage site, made a site mutation, so this is no longer cleaved. We get a stabilized version of CD16, and with our collaborators at Fate Therapeutics, they made a version of this that also expresses the high affinity isotype of CD16, but this occurs in about 10% of people, which have an improved affinity for antibodies. So we expressed this so-called high affinity non-cleavable CD16 in iPS-derived NK cells, and this just shows compared to the unmodified cells or peripheral blood NK cells, which normally express CD16, when, for example, on the bottom, when the peripheral blood NK cells get activated, they either by tumor cells in the middle or by pharmacologic agonists, they lose much or all of their CD16. However, the HN-CD16 NK cells maintain high levels of CD16 and improved, in this case, cytokine production when they're stimulated. And so we tested this initially in vitro, and we could show, again, with 3 different antibodies in either a short-term assay in the top with a CD20 positive leukemia cell plus rituximab. Bottom left is with cetuximab, and bottom right is with Herceptin. Those are longer-term assays, and again, the purple in all of these shows better anti-tumor activity with the so-called HNCD16 iPS-derived NK cells. This is in vitro. We've now tested this in vivo, again, against a B-cell leukemia model combined with Rituxan. Again, the antibody alone does give some anti-tumor activity. We do this with the unmodified iPS NK cells or peripheral blood NK cells combined with antibody. Again, we get improved anti-tumor activity and improved survival. But when we did this with the iPS-derived NK cells with the stabilized CD16 plus anti-CD20 antibody, we again got better survival and 3 out of 5 mice with long-term survival. So we could actually show in this lymphoma model, and now doing this with other models, that again, by having this more stabilized CD16 expression, we can mediate improved anti-tumor activity in vivo. The last thing that we did is we wanted to show we could not only overexpress or alter the receptors on the surface of the cells, but we can also genetically modify the iPS NK cells to make internal gene edits to knock out so-called internal checkpoints. And so the one we focused on this gene called CIS. The protein is called cytokine-inducible SH2 containing protein. I had to write that down to remember it. But this is actually involved downstream of IL-15 signaling. So IL-15, again, is an important activating cytokine for NK cells and cytotoxic T cells. However, this protein CIS normally turns off that signaling by leading to ubiquitination of JAK and STAT downstream signaling molecules. This leads them to be degraded. So if you knock out CIS, you maintain the activation through IL-15 longer. And this was shown in CIS-deficient mice that improved T cell and NK cell activity and improved activity against tumor cells in the mouse model. Nobody had really looked at this in the human models, so we knocked out CIS in our iPS-derived NK cells using the CRISPR-Cas9 system. I took out the slides, but this didn't really impact the development of NK cells. You get normal phenotypic mature iPS-derived NK cells. We showed that these could actually proliferate better at low concentrations of IL-15, so this is normally part of the cytokines that we have to grow these. If we reduce the dose to just 5 nanograms per mil, normal NK cells won't survive, but these CIS knockout cells will survive. We've now tested these in vivo, again using a leukemia model, IV injected leukemia tumor cells, 1 IV injection of the cis knockout cells, which again in the right shows improved anti-tumor activity, and I don't know if showed here that this corresponds to improved proliferation and survival of these cis-deficient iPSNK cells in the blood of these mice. And we know from all the trials of NK cells and CAR T cells, the longer your cells survive and proliferate in patients, the better the anti-tumor activity. And so because these cis knockout cells can survive better under low cytokine concentrations, we get better survival and persistence, and that corresponds to better anti-tumor activity. So really, just to summarize, that we can make various versions of iPS-derived NK cells, either with CARs or with other gene modifications. This really can be a standardized off-the-shelf therapy. They don't need to be patient matched. These can be done as a centralized manufacturing. You do get persistence of the cells, but you also don't get the same lifelong potentially persistence that you do with CAR T cells. This can be a positive or negative, but we can redose with the NK cells and give a more precise dosing. They seem to have less toxicity than CAR T cells. As I mentioned, we can do repeat dosing, and we can combine these cell engineering strategies. So I didn't put up here, but again, in collaboration with Fate Therapeutics, they've now started the first clinical trial of iPS-derived NK cells, so-called FT500. This is an unmodified iPS-NK cells that's just started in the last couple months in trials. Really too early to say anything about those. But they also now have an IND approved for the second iPS-derived NK cell, which will express that hnCD16. That'll be combined with antibodies, again, to treat patients with refractory solid tumors. So they not only have the first IND for iPS-derived any type of cell in the U.S., this is the first iPS-derived blood cell and the first iPS-derived cell to treat cancer anywhere in the world. Also, with the subsequent trial, will be the first engineered iPS-NK cell, first engineered iPS cell at all, which will be an NK cell. Their third product then is going to be the CAR-expressing NK cells, actually combined with the CD16, because we can combine modalities. So that'll be anti-CD19 with the hnCD16 can then be combined with the antibodies to prevent antigen escape and provide improved activity. So those are in their pipeline. So this is the people in my group who I get to take credit for all the hard work that they do. Really the ES and iPS-derived NK cells we've been working on for a decade or more. As I mentioned, Petter Wohl, David Noor, David Hermanson really pioneered that work. The CAR NK cells was done largely by Ethan Lee, and Huang Zhu is a current postdoc who's been doing both the CD16 and the cis knockout work. And then valuable collaborators both at University of Minnesota and Fate Therapeutics. Really has been a great group to work on, and really excited to see how this will progress. So I'll stop there. Thank you.
Thank you, Dave. We may have the time for 1 to 2 questions. Please go to the microphone, please.
Great data there. My question is, did you see any teratomas in the mice infused with these, and how are you testing for residual iPSCs in your final product?
Right. So when we do this in mice, and I mean we've injected, mice with a variety of ES and iPS products. These are very differentiated cells, and so the short answer is no. We don't see teratomas in these. I don't have privilege to all the data that was done for the IND, but I know, that those sort of dose-finding studies need to be done for the IND submission. And so really you can show that there's no residual undifferentiated cells in this product, and we don't get teratoma formation. I don't have all the details, but that's the yeah.
I was wondering if you could speak a little bit about the clinical differences between knocking out the negative regulator of IL-15 versus Katie Rezvani's approach of inserting constitutive IL-15, because these cells will still be dependent on some external source of IL-15, and when people are preconditioned, do they have that, and at what level?
Sure. That's a good question. And we haven't actually compared it head to head. So I can say, again, and I'm limited to say things that are publicly available if you look at Fate Therapeutics' website, that third product will also actually express a CD15 receptor fusion molecule to provide the exogenous sort of stimulus. So we haven't compared head to head. So CIS, does affect other signaling pathways as well, and there may be sort of an added benefit. It does require systemic cytokines, but as you probably know, you get sort of a homeostatic increase in cytokine levels when patients get lymphodepleted. So these cells are given with cytoxin, fludarabine conditioning, suppresses the endogenous immune cells, and you get sort of a corresponding increase in IL-15 and other cytokines. And that's for sort of that period of lymphopenia. So how that compares for these CIS knockouts versus the IL-15, it's actually an interesting experiment. I think we might have to do that, but we don't know firsthand. So,
Okay.
thanks.
Thank you, Dan.
Okay, thank you.
Thank you.